Hey there, my fine specimens! In this video, as promised, we will be talking about ANAs. In particular, interpretation and correlation of ANA patterns is one of the most commonly asked topics in the ASCP exam. I can attest to that because during my examination, there were about 3 to 5 questions related to this topic. So I hope that this video could really help you out during your review.
As what you may already know, ANAs stand for anti-nuclear antibodies. Obviously, by the term itself, this refers to antibodies that targets the cell's nucleus. ANAs are classified as autoantibodies since they target the body's own cellular nucleus and components. These components include nuclear proteins such as that of histones, nucleic acids, of course, the DNA and the RNA, and nucleic acid protein complexes. These complexes basically refer to DNA or RNA that are bound or attached to nuclear proteins, examples of which include RNPs and DNPs. Just a quick reminder, although we use the terms ANA and ANAs interchangeably, there's quite a difference between these two terms. ANA is more of a general term that refers to a non-specific antibody that binds to the cell's nucleus. On the other hand, ANAs are a group of different anti-nuclear antibodies or a heterogeneous group of antibodies that bind to particular or specific nuclear components. Now, to make things easier for me during my review, and I hope this could help you too, I have classified ANAs into 3 different types. This is based on the specific nuclear component that they target. Now, this is helpful because this same classification can be applied to the type of the immunofluorescence pattern that each of these antibodies will exhibit during immunofluorescence testing. So the first type of ANAs are those that targets the DNA or the DNA protein complexes. This includes your anti-dsDNA, anti-ssDNA, anti-DNP, and anti-histones. The second type are those that targets non-histone nuclear proteins. So this refers to all nuclear proteins except your histones. And they could also target your RNA protein complexes. So this type includes anti-U1RNP, anti-Smith, anti-SSA, anti-SSB, anti-centromere, anti-SCL70, and your anti-Jo1. And the last type of ANAs include those that targets nucleolar antigens. Of course, these refers to antigens or proteins that are found within the nucleolus. This includes your anti-RNA polymerase 1, anti-U3RNP, and your anti-PMSCL. Testing and identification of these anti-nuclear antibodies aids in the diagnosis of autoimmune diseases associated to connective tissues, such as SLE, Sjögren's syndrome, systemic sclerosis, and polymyositis. But it is of utmost importance to remember that positive ANA test result does not always indicate an autoimmune disease, because 20 to 30% of the healthy population can actually test positive for the presence of ANAs. ANA testing is therefore significant in ruling out autoimmune diseases for individuals who will show negative ANA test results. Therefore, for us to have an accurate diagnosis, it requires the proper interpretation of the patient's history, in addition to the manifestation of clinical signs and symptoms that correlates to a positive ANA test result.
Though there are different methods for ANA testing, we'll only be focused on the most widely used method, which is the indirect immunofluorescence assay. This is primarily used as a screening test for the diagnosis of SLE and other systemic rheumatic diseases, and that is because of the test's high sensitivity. This means that it is able to detect presence of anti-nuclear antibodies even in small amounts. Thus, a negative result will most often rule out or exclude SLE and other systemic rheumatic diseases. However, this test exhibits low specificity because the substrate utilized does not represent a specific component of a nucleus, but rather it represents the nucleus as a whole. So in this method, the patient's serum is introduced into a well that contains our substrate. The substrate used can either be a mouse or rat liver or kidney cells, or cell culture such as hep2 cells can also be utilized. Then, if the ANA is present in the patient's serum, it will bind to the substrate. The next step is to wash it with buffer in order to remove unbound antibodies to avoid test interference. Then, the mixture is incubated with fluorescein labeled anti-human globulin, which will then bind to the ANA substrate complex. Again, we will be washing this with buffer to remove unbound antibodies. Once viewed through the microscope under UV light, a green or gold fluorescence will be observed in the presence of ANAs. And take note, distinct immunofluorescence patterns can then be correlated as to the type of ANA present in the patient's serum. As I mentioned earlier, the three types of ANAs can be correlated to distinct immunofluorescence pattern. For instance, homogeneous and peripheral immunofluorescence pattern usually indicates the presence of the ANAs that target the DNA or the DNA protein complexes. Speckle patterns, on the other hand, usually indicate the ANAs that target the non-histone nuclear proteins and RNA protein complexes. And of course, nucleolar immunofluorescence patterns indicates the presence of the ANAs that targets the nucleolar antigens. So let us now dive into the different ANA patterns and the particular antibodies and diseases associated to each of them. First is the homogeneous pattern, also known as the solid or diffuse pattern. Again, this is associated to the ANAs that target the DNA and DNA protein complex, particularly your anti-dsDNA, anti-ssDNA, anti-DNP, and antihistones. So for anti-dsDNA, of course, it specifically targets the double-stranded DNA, also known as the native DNA. Another particular characteristic of anti-dsDNA is that it is able to bind with the circular DNA found in the kinetoplast of a hemoflagellate called the Crithidia luciliae, which is why Crithidia luciliae immunofluorescence testing is considered as the gold standard for the specific identification of anti-dsDNA. Next, for anti-ssDNA, of course, it targets your single-stranded DNA, also known as the denatured DNA. For anti-DNP, it targets your DNP or the deoxyribonucleoproteins. These are again the DNA bound to nuclear proteins, example of which is your nucleosomes. And lastly, we have the antihistones that targets, of course, the histone proteins, which may include one of the following. So the H1, H2A, H2B, H3, or H4 histone core proteins. Now, high titers of these homogeneous ANAs are associated with SLE, although they could also be present in other diseases such as rheumatoid arthritis, mixed connective tissue diseases, and Sjogren's syndrome. In particular, the anti-dsDNA is considered as one of the specific markers for SLE, as they are seen in 60% of patients with SLE. For antihistones, if they are solely identified or present, they are associated with what we call as the drug-induced SLE. Drug-induced because this usually occurs in the presence of drugs such as procainamide, an antiarrhythmic drug, and hydralazine, which is a vasodilator. So in the presence of these drugs, the patient may manifest SLE-like symptoms. Under the microscope, this is how the homogeneous immunofluorescence pattern look like. So another tip, guys, is for you to draw on your own these immunofluorescence pattern, because it is a big advantage if you are familiar with how these patterns look like, because during the ASCP exam, it's usually that images of the patterns are being shown, and then of course the question would be related to that particular image. So for homogeneous pattern, the nucleus evenly fluoresces, as what we see here in the picture. Next, we have the peripheral pattern, also known as the rim or the marginal pattern. So the antibodies associated to this pattern are only the anti-dsDNA and anti-DNP. So we have already discussed their specific target nuclear components. As for the diseases associated, so these peripheral ANAs are seen particularly in SLE, and also they are seen in cases of Turgeon syndrome. In the case of your peripheral pattern, as what you could see here, the central portion of the nucleus is only lightly stained or no staining at all is present, but there is a strong fluorescence at the nuclear margins or at the periphery of our nucleus. For speckled patterns, there are actually various types, but first we have here the core speckled pattern. So associated to this are your anti-Smith and the anti-U1RNP antibodies. Now remember guys that these two usually occur together because they target different antigenic sites of the same protein structure, which is your U1 small nuclear RNP. Specifically, your anti-Smith will target the Smith protein complex or the Smith antigens, which are made up of 7 protein structures, while your anti-U1RNP will target other proteins such as that of your RNPA protein, RNPC, or the RNP70KD protein. So for both anti-Smith and anti-U1RNP, they are seen in cases of SLE. In particular, anti-Smith antibodies is considered as one of the most specific markers for your SLE because this is usually seen in SLE alone, and this is detected in 20 to 30% of patients with SLE. Anti-U1RNP, on the other hand, can be seen in other diseases such as the mixed connective tissue disease or MCTD, systemic sclerosis, the diffuse type, and Turgeon syndrome. In particular, anti-U1RNP is seen in high titers in cases of mixed connective tissue disease. For coarse speckled patterns, so here is the appearance. So they have this coarse, grainy, or granular appearance without staining in the nucleolar area. Next is the fine speckled pattern. Associated to this is the anti-SSA-RO and anti-SSB-LA. So both of these antibodies are directed towards the antibodies found in the ROLA-RNP complex. In particular, your anti-SSA-RO targets your RO proteins, which could either be your RO52 or RO60, while your anti-SSB targets the LA phosphoprotein of the same complex. Both of these antibodies exhibit high titers in cases of Turgeon syndrome. Between the two, anti-SSB is present in a higher prevalence, as this is seen in 60% of patients with Turgeon syndrome compared to anti-SSA, which is seen in 40% of patients with the same disease. In lower titers, they could also be associated to SLE. So for fine speckled pattern, it is somewhat similar to the coarse speckled pattern, but as what you could observe here, the grainy appearance is finer. Next, we have the discrete speckled pattern associated with your anti-centromere antibodies. So the anti-centromere antibodies targets the centromeric chromatin, and this is particularly associated with systemic sclerosis, the limited cutaneous type, particularly for those patients exhibiting the CREST syndrome. So what is this CREST syndrome? C stands for calcinosis, which is associated to calcium deposits in the skin. We also have the Raynaud's phenomenon associated with the spasm of small blood vessels, particularly of the fingers, that may cause skin discoloration. Next, we have esophageal dysfunction, which could result to acid reflux and decreased motility along the esophagus. We also have sclerodactyly associated to the thickening and tightening of the skin, particularly of the fingers, which gives the hands of the affected individual a claw-like appearance. Lastly, we have telangiectasia associated to the dilation of small blood vessels, particularly seen in the face. So this is how a discrete speckled immunofluorescence pattern look like. So for its description, it is associated with discrete coarse speckled granules which could either be scattered in interphase cells or they could be aligned in metaphase cells. Next, we have the typical speckled pattern associated to the presence of your anti-SCL70 autoantibody. So this antibody targets the DNA topoisomerase 1 and is associated with individuals who have systemic sclerosis, the diffuse cutaneous type. But this could also be seen in lower titers in cases of SLE. So for a typical speckled pattern, it is described to have the same appearance of your fine speckled pattern. What makes it atypical is the uneven distribution of immunofluorescence. The last type of speckled pattern is the cytoplasmic fine speckled pattern. So this is associated with the anti-Jo1 antibody that specifically targets the histidyl tRNA synthetase. So this antibody is associated with polymyositis but could also be seen in cases of dermatomyositis. This is how a cytoplasmic fine speckled pattern looks like. So you might be confused. It is considered as an anti-nuclear antibody, but why does it exhibit immunofluorescence, particularly in the cytoplasm? So that is because the histidyl tRNA synthetase, which is targeted by our anti-Jo1 antibody, could be seen in both the nucleus and the cytoplasm. So for this pattern, they are described as having that small scattered speckles in the cytoplasm which appears even or homogeneous. Lastly, we have the nucleolar immunofluorescence patterns. So these are again associated to antibodies that targets nucleolar antigens that includes your anti-RNA polymerase 1, anti-U3RNP, and anti-PMSCL. So for anti-RNA polymerase 1, this targets, of course, your RNA polymerase. Anti-U3RNP targets, on the other hand, your U3 small nuclear RNP. So for these autoantibodies, they are primarily associated to a polymyositis sclerosis overlap. As for the sclerosis, they are usually associated with the diffuse cutaneous type. So for the nucleolar pattern, this is how it looks like. So as what you could see, there are few round nucleolus that stains within the nucleus. So that's it for ANA testing. As what I have mentioned, this topic repeatedly came out during my ASCP exam. So I suggest you guys study your ANAs. So I hope this video could be of help not just for those who are about to take the ASCP exam, but also to those medical technology students or interns that are studying for the local board exams, your revalidas, or even just with your daily quizzes. You can show your support by clicking the like button. You can also comment down below any suggestions for our next topics, and of course hit subscribe button guys for more. You could also follow and support us through our Facebook page at Acamazing Medtech. See you again, fine specimens!